Okay, so good job getting test 2 done. Test 1 done. Now we are on to test 2. I think, if I'm not making things up, test 2 just closed on Tuesday, so it can take up to a week to grade practical exams because they are fill in the blank and I have to like manually grade them and I have to review all of that respondus footage. So hopefully I'll get it all done today and tomorrow. When the grades are posted, be sure to check your feedback, and that's a good habit to get into all the time. Any assignments that you've turned in, check for comments from me, because a lot of times I'm telling you what you need to fix in order to get like a higher score on the assignment. Okay. For tests, I'll just give you feedback about like, just whatever I think might help for the next test. But if you want to review your test, you have to come to office hours. So once it's graded, if you want to like go over what you missed, that's an office hours thing to do. So check out the office hours module to figure out how to get to office hours and when they are. Okay, so test 2, we're going to start moving into bigger and more complex organisms. Okay, so what have we already done? We did prokaryotes, which was 2 whole domains of life. Then we got into protists. Those are the first eukaryotes we met. And now we're going to meet a lineage of organisms that evolved from those protists. Specifically, plants came from green algae. Okay, so we have a green algae ancestor. And there are a couple of reasons why we theorize that green algae is where plants come from. So what is something that you know about green algae and plants that they have in common? What are some green algae plant similarities? Photosynthesis, very good. Okay, so photosynthesis, being able to make food out of sunlight and carbon dioxide, is not something plants figured out on their own. That is something they inherited from their green algae ancestors. So photosynthesis. And green algae aren't the only photosynthetic organisms, right? We've got different kinds. We've got brown algae, red algae, golden algae. These are all not as closely related to plants, but green algae are. Yes, cellulose was a good thing. So chloroplasts, I'm just going to fill in that cell wall of cellulose. Green algae had that. Plants have that too. Chloroplasts, specifically chloroplasts with chlorophyll A and B, which are the green pigments in chloroplasts. That's why plants are green. Okay, so whenever we talk about plants, the kingdom Plantae, we're referring to land plants. They're all terrestrial. And they're all green. Okay, they're all green because they have these chloroplasts. Okay. So these are all things that plants didn't invent. Okay, I've got things up here that are not derived. These are ancestral traits up here, things that they inherited from the green algae. Everything that we're going to learn going forward is what makes plants adapted to their environment. Okay, so try and think of like, what are the challenges that a plant is going to face? Okay, because where do green algae live? They live in aquatic environments, oceans, rivers, streams, stuff like that. Okay, so the green algae ancestor moved on to land, okay, and became land plants. So the land didn't have a lot of living things on it. It had fungi, prokaryotes, but no plants or animals yet. So the first algae ancestors started moving on to plants. And that's what we're going to start with, things that more closely resemble algae in a lot of ways. So mosses, liverworts, hornworts. These are avascular. They don't have vascular tissue, which allows them to grow taller, like ferns and trees and plants that we're used to. So these are going to be short, low, shallow plants that can just barely, they have to be in a moist environment. Okay, they're only a little bit adapted to land. Okay, so that's our first lineage. Okay, we're going to start with them. And then every step up this plant group tree gets more and more terrestrial. Okay, they are adapted to terrestrial environments. Terrestrial just means on the land, like not aquatic. Aquatic is in the water, terrestrial is on land. So what are some challenges that an organism might face if it's moving from water to land? What are some problems with living on land? Think about if anyone's ever been like up north where it's colder and drier, what happens to you? What happens to your skin and stuff? Yeah, drying out. Yeah, perfect. So you get it, you lose water to your environment. All of our cells are filled with water. So you lose water to your environment. You have to have some way of making sure that your cells are always getting what they need and they're moist enough and you have, you know, enough water to do the chemical reactions that cells need. Okay, so if you live in the water, you've got water, you've got everything you need just floating around. Okay, gases are just dissolved in the water. Ions that you need, compounds, they're just floating past and you just have to be able to get them. So algae did that. Moving to the land, you had to think of ways to not dry out. So one of the first adaptations that evolved and allowed these plants to live would be a waxy cuticle. Okay, we're going to see that on leaves and things. But that prevents the cells from drying out. So plants are truly multicellular. Okay, that's something under what is a plant. Okay. Photosynthetic, these are all things that make a plant. By definition, plants are multicellular. There are some weird plants that like don't do photosynthesis. They've lost it. Like maybe they become parasites. We've seen that with protists as well. Things that change lifestyles can lose certain functions that they once had. So that's an evolutionary reversal or a loss. Okay. So that waxy cuticle, that's like the main thing that mosses and their friends have. So they don't dry out. They can survive a no water situation. Okay, so it's hypothesized that the first land plants were like mossy things that lived on rocks around like tidal areas. So they would sometimes be submerged in water, but they could survive low tide without like dying. Okay, so that was kind of the first step onto land. And then it just, you know, things get better and better at living where they won't dry out. Okay, there's also the problem of getting water. If you're living on land and you're not living in a pond or a constant puddle, if water is not always available, you have to find ways of getting water. And where is water all the time? Pretty much, like even if it hasn't rained for a while, there should be water somewhere in the environment. Where is it? Yeah, soil, right, soil, air. These are places where plants can access water, even though they're not like in a pool of water. So where is the water in the soil? It's usually down kind of deep, depends. Like New Orleans is pretty soggy. You might not have to dig very far to get water. But there's different, you know, like there are orchids and succulent plants that can just get moisture out of the air. Okay, in like really tropical environments, you've got plants growing on walls and on power lines because they just have enough moisture available to them. In drier environments, colder environments, the water might be further down, and you need to grow structures that get down there. Okay, so I'm just going to go over to my little roots and shoots image. Okay. Oh, no. Okay, so roots and shoots. This could not exist without vascular tissue. So that's the next big advancement in plant evolutionary history, or plant evolution. Okay, vascular tissue. Okay, so vascular tissue allowed for plants to grow taller. Okay, they could grow towards where their sunlight is. They really need sunlight. But they can also grow roots down. So mosses evolved before vascular tissue. Okay, this lineage split off before vascular tissue existed. So mosses are going to be shallow, and they're just going to have to get the water from what they're sitting on top of. Okay, so let me show you this model of a moss plant. Sorry. Okay. Here. Okay, so here are some images in your PowerPoint. This leafy structure here, it's not a true leaf, but it's the body of a moss. Okay. Mosses don't get super tall. Okay, this is a very tiny flat thing. It's low to the ground. And they have these things that kind of look like roots. Okay, it's called a rhizoid. Rhizo means root, rhizoid means root-like. So it's not a true root because true roots have vascular tissue. You got to think back to 143, plant anatomy. You can look at the cross-section of a root and see the vascular phloem and xylem. Okay, so that's a lot of cells and tissues to make a structure. Here we just have like a little row of cells, and they're sucking up water into their insides of the cells, and they can pass that water to other cells, but it's all through diffusion. It's kind of not that efficient. Okay, it's not like just a straw sucking up water, which is what vascular tissue does. So mosses are land adapted, but not quite. They're still very dependent on having a moist environment. They can't get too super tall, so the next plants that evolved to grow taller could shade out these mosses and compete with them. Okay, so the taller you get, the more things you can do, the more environments you can exploit. But we're going to appreciate the mosses first. We're going to start with them, learn their life cycle, learn all their different bits and pieces, but appreciate that they are still very much tied to water. We're going to see parallels when we learn animals, right? Animals started in the ocean, you got fishes and things, and then some fish ancestor moved onto land. And then we see successive adaptations to living on land in amphibians and reptiles and mammals. Okay, so plants are going to do the same thing. Mosses are a little bit terrestrially adapted, but they still depend on water for most of their life cycle, especially reproduction. So sexual reproduction, we think about sexual reproduction in the, okay, let me come back to that real quick. I have a question in the chat that I will come to, but let's talk about moss reproduction. Okay, moss and many plants can reproduce asexually, but not all. Okay, a lot of them can do sexual reproduction, and that means you have sperm and eggs, and sperm have to go find the eggs. So if you're in water, that's not that hard. The sperm can swim and find the egg. On land, it's not that easy. Okay, so later plants are going to be even better at getting sperm to eggs without drying out, or they can dry out and it doesn't matter. Mosses can't do that. They still need a little bit of moisture for the sperm to swim. They still have flagellated sperm that just swim from the male parts of the plant to a female part where there's an egg cell, and then fertilization happens there. Okay, so they're still really tied to water. The next group that we're going to talk about is ferns. Okay, ferns and things that are related to ferns. And then we're going to see next week the seed plants and see how the evolution of seeds and pollen kind of really changed the game. So I have a question about why does moss disappear when it's dry and then reappear when they are wet? I'm not sure that disappear is the right word. I'm not really aware of this phenomenon. They could dry up and then come back, but they could die away from that spot and then grow back when conditions are better, but I'm not exactly sure about that. So let's take a look at the module real quick because I want to point out some things. So you should have already done the pre-lab stuff. If you haven't, just start there. A lot of pre-lab information, and we're going to talk about the weird life cycle thing real soon. But yes, good. So what's going on here for the exercise? The pre-lab is normal. It just asks you questions about what's in the pre-lab handout. We'll talk about population genetics before the end of the lab. This is your activity. Okay, the seedless plant life cycles assignment is like one part of what you're going to turn in, but on top of that, you've got just the normal exercise where you have images in a PowerPoint. Okay, the images, and then you go along with the chart and the exercise and identify each picture, kind of put it into context of taxonomy, what's the deal, where does this thing live, how does it reproduce, all that stuff. Okay, so this is kind of like traditional 144 lab module stuff here. And then you're going to have this more interactive activity that you have to do in PowerPoint. You might be able to do it in Google Sheets or something, or Google Slides, but you have to turn in a PowerPoint. So be sure to get Microsoft Office. There is a free download for that. So you need to be able to use certain programs. That's part of this class. Okay, so you're going to use a PowerPoint to place the images. So a lot of the images are going to come straight from your PowerPoint, and you're going to put it onto the life cycle of plants. So we're going to learn the general life cycle of plants, and then you're going to look at mosses and bryophytes and say, okay, here's an example of this generation. Here's an example of the haploid structures. Here's an example of the diploid structures. Okay, so it's like putting each individual plant life stage onto the general plant life cycle, and then you're going to turn that in. So there's a moss life cycle and a fern life cycle, and all you're going to do is move images around and label those images. So you're going to use the PowerPoint information and the pre-lab handout information to arrange things and kind of make sense of it. Okay, so let's talk about that life cycle. Just go, I've already drawn this like a million times. So we've got some weird words. Okay, plant life cycle, plants are weird. Let's think about human life cycles real quick. What is fertilization?
When the sperm is with the egg.
Yeah, sperm plus an egg. Okay, that makes a zygote. And that turns into, we're gonna skip some steps, a baby person, right? Okay, that baby will turn into an adult. That adult will produce sperm or egg, some kind of gamete. And in order to get back to the life cycle, we need the other one, right? So another gamete joins together, and that's fertilization. Okay, sperm plus egg. Okay, so we can make human life cycles seem complicated, just like we can do with plants. Okay, now let's start applying some of these super fun biology words. me just make it make a little bit more sense. Okay. Okay. So haploid and diploid. Okay. Which of these things are haploid? And let's just start with haploid. What are some things that are haploid on this list? Is a baby human haploid? Yeah, sperm and eggs. Okay. So we're gonna say sperm and egg gametes are haploid. Okay. These are just single cells. Okay. They're not that, these are not multicellular haploid structures. We just have only these specialized cells are haploid. Everything else is diploid. Okay. A zygote, baby, adult, they're all diploid. We can also write that as 2N, haploid is N. What does that mean? That means how many chromosomes they have. A sperm has half the amount of genetic information needed to make a new individual. The egg has the other half. So for humans, we've got 23, chromosomes. 23 is the number of different chromosomes we have, but we've got 2 pairs of each. We have 2 of each of those 23. So in humans, N is 23, 2N is 46. 2 times 23 is 46. Okay, so chromosome 1 through 23 comes from one parent, and another whole set of 1 through 23 comes from the other parent. And they go together to make a full diploid individual. The word for this, and this is animals, we're diplontic. Okay. This, from here to here to here, this is all multicellular. Okay, that's all multicellular. The only haploid cells that we have are individual gametes, single cells. Okay, plants are super weird. Instead of just having sperm and eggs come from the adult plant, we have separate haploid structures that produce the gametes. Okay, this is what's weird. Instead of being diplontic, plants are haplodiplontic. Okay, so let me draw them a little person again for comparison. In a person, all of their cells are diploid. The only cells that are haploid are going to be located in the gonads. So just the cells that turn into sperm and eggs are going to be haploid. How do we get haploid cells? We do meiosis. Okay, so meiosis is how we get from a diploid cell. Okay, so one of these adult cells is diploid, and meiosis does 2 rounds of cell division to get a haploid. Okay, fertilization takes 2 haploid cells and makes 1 diploid cell. Okay, it's going to be the same for plants. We're just going to add some steps. Okay, I'm going to try and make it neater than what I have done here. Okay, so we're going to come up with all these new words. We've already got haploid and diploid. Sorry, we just have to do haploid and diploid. But we're going to add some much worse words. Okay, so we've got gametes. We already kind of know what those are. Gametes are sperm and eggs. Okay, these are both haploid. And they have to fuse together in an event called fertilization. Okay, and that makes a zygote. So far, that's normal, same as in humans, same as in animals. Okay, but what produces the gametes is a multicellular weirdo structure that plants have called a gametophyte. Okay, this is a haploid structure. So it doesn't have to do meiosis, it just makes gametes via mitosis. Regular, just like it's already got one, let's say N equals 16. There's 16 chromosomes. In the diploid, they would have 32. Here, the gametophyte cells already have half the information. So they just make a copy, they just split into two, and those daughter cells are gametes. They're already haploid. Okay, so that's the weird thing. That's what's weird about plants. They have this gametophyte generation. We call it a generation, and it is multicellular. Okay, so it's like a little multicellular structure. Let's go from the zygote all the way to the gametophyte, and then we're going to talk about how this is the same for most plants, but then what's different between plants. Okay, so we're going to draw a line. Okay, so up here, this is all N, and below the line is all diploid. Okay, we have a haploid generation and a diploid generation. So we know how to get from haploid to diploid. These gametes are haploid, we get a 2N zygote. Fertilization is that process. Okay, so the zygote grows up, it divides by mitosis to make a multicellular sporophyte generation. Okay, so if you think of a rose bush is the sporophyte generation. A pine tree is the sporophyte generation. Mosses, the moss you think of when you think of a moss, or the moss we just looked at, that structure is the gametophyte. Okay, the little flat kind of just leafy thing on the ground, that's the moss gametophyte. In mosses, we got that thallus, it grows this little sporophyte. Okay, a little stalk thing. This is the sporophyte. Okay, sporophyte releases spores. Okay, we're going to have special structures called sporangia. And they're going to be located in different places, but let's say they're in here. Okay, the sporangia inside there, they do meiosis. Those cells do 2 rounds of cell division. These cells are still all diploid, diploid, diploid. Okay, when we do meiosis, then you get haploid spores. Okay, those spores grow up, they divide by mitosis into the gametophyte generation. Okay, so these are all crazy words. This is all new. You probably won't need this information tomorrow at work. This is really weird plant life cycle stuff, but it's really important to how plants work and going through the different groups. So we're going to learn 4 different phyla of plants, pretty much, or 4 different main groups. And then we'll see how this weird life cycle changes as we evolve and adapt to living on land. Okay, so for mosses, the gametophyte is the main plant, and the sporophyte is just this brief thing that happens when reproduction needs to happen. Okay, when it's time to make spores, they do this little fertilization, sporophyte, make spores. Okay, but the biggest part of a moss life cycle is going to be that gametophyte. That's what does photosynthesis. Okay, that's kind of interesting. The gametophyte generation is the one that's living its normal life and taking in sunlight and making sugars and doing its normal plant thing. Okay, in ferns, when we go up to the next level of complexity, or the more adapted to land, those ferns, the sporophyte generation is going to be what you think of, like the fern that you think of, that you would have on your porch, or if you see ferns used in landscaping, what you're thinking of is the sporophyte generation. And that's going to be true for all the rest of the plants. Sporophyte becomes the dominant generation. That's what does photosynthesis. And the gametophyte gets reduced. So mosses start with a big gametophyte, and then by the time we get to seed plants, those gametophytes are going to be almost microscopic. Okay, they're just going to be part of the adult sporophyte plant. Okay, so try and pay attention to that as you go. This won't really make any sense before you start delving into the different examples and paying attention to what part of the life cycle you're looking at. Okay, so some plants you should already be familiar with. What you want to do is relate the things you're familiar with to this kind of weird alien life cycle that we're learning about, okay? So the activity today is going to have you place, like once you've gone through all the images in the Seedless Plants PowerPoint, and you kind of connect the dots between the image and the part of the life cycle that it represents or it's a part of, then it'll be easy to do that activity. So just kind of go through all the exercise, keeping in mind this weird gametophyte, sporophyte thing, okay? Definitely practice just drawing this from memory, okay? You've got the meiosis and fertilization on either side, gametophyte and sporophyte, and then just kind of make sure you understand what's happening from one stage to the other, okay? Haploid and diploid, like you just have to accept these words and learn them for now, and then you can put them away until you need them again, but you just have to like get through it. Gametophyte and sporophyte. The easy way to remember it is gametophytes make gametes, and then the sporophyte makes spores. The only thing that's tricky about that is sporophyte is diploid, but spores are haploid, because to get from the sporophyte to the spores, you do meiosis. So just like practice that. Maybe once a day, try and draw the whole thing from memory and check your answers. And if you have this alternation of generations life cycle just sort of in your brain, it's not as hard to think of these things under pressure, okay? So I'm gonna let y'all have some time with the module information, okay? So just start with the exercise, work your way through, like use the exercise to follow along the PowerPoint. And when you get done with that, start on the life cycle assignment, okay? And I can help you with the, like fighting with PowerPoint. It is annoying. Sometimes things will be on top of other things. Like there's little tips and tricks to make that work, but you've got everything you need in this PowerPoint. You just have to arrange it correctly, okay? But before you do that, I want you to look through all of these, like just go through the exercise sheet first, okay? So I'm gonna stop there, and then we're gonna just work on stuff. I'll let you work in the breakout rooms. It's 7:30 now. So you don't have to watch these videos. They're just there for more help. If you're struggling with it, you can watch a video. But we can, you know, I'm in class now, so we can talk about this. That's like if you're absent or something. But so let's talk more about plants at like 8 o'clock. Okay, so I'll let y'all work for half an hour and come back, and we'll talk a little bit more about mosses, and then move into ferns, give you some like fun fern details, and we'll talk a little bit more about life cycles. And then a little bit later in the period, we're going to talk about population genetics so that you're able to work on that. Okay, any questions before we begin? We'll do an 8 p.m. check-in, and then probably a 9 p.m. checkout. You're gonna start up here and then just work your way to this one. All right, no questions. I'm gonna open breakout rooms. If you wanna sign out and just come back in at 8, that works too. But if y'all wanna chat with each other, I think that always makes it a little bit more fun. Breakout rooms are open, and I'm here for questions if you run into any trouble. Record again. So I looked over the pre-lab quizzes. So not everyone has completed that. If you haven't, be sure to do that, you know, so that you can progress into the activity and see what you're getting into for today's activity before it's too late. So if you have questions, I can help. But that pre-lab is a little tricky, you know, like those questions that ask you to choose all the haploid structures or all the structures produced by mitosis. A quick little help with that If you didn't get a 10 out of 10, try again, okay? But let me just kind of point out some things that are easy to miss or to not put together at first. So I can't really put this information in your brain, okay? Like you have to struggle with it and learn it yourself, but I'm gonna try and help as best I can because I know this is really weird. If you look here, all of these arrows are labeled mitosis, okay? Remember, mitosis is just making a copy of the cell, right? So whatever the parent cell has, that's what the daughter cells get, okay? So you can use this to make a single-celled thing into multicellular things, okay? Like when an egg and a sperm fuse, you have that single-cell zygote. To grow up into a mature individual, that zygote cell divides into two, those two divide into two, and you just keep doing mitosis until you get trillions of cells, right, that make up a whole organism. Okay, so all of these arrows that go from a stage, like we're going from spores to gametophyte. You don't have to do anything special, you just do mitosis. So one spore divides by mitosis to grow into a multicellular gametophyte. Okay, going from a gametophyte to an archegonium. Okay, that's just, again, more mitosis, just growing a new structure. Okay, mitosis is just making copies of cells, but cells are fancy and they can grow into different shapes and make different tissues and stuff like that. So all these, like this arrow to this, okay, from here to here, from here to here, everything, when you're staying above the line, it's all mitosis and it's all staying haploid. You start haploid and you end haploid. Haploid to haploid, haploid to haploid, okay? Same thing with underneath the line. An arrow that goes between stages in the diploid phase, it's always gonna stay diploid and you're gonna do that from mitosis. So zygote to sporophyte, just divide by mitosis, okay? To get from diploid to haploid, right? We got 2 in, to make half the information, that's meiosis. Just like in sexual reproduction in animals, meiosis takes a diploid cell and splits it into haploid gametes. Plants just take the long way around and they make spores first. Spores make a gametophyte, and so on. Okay, so meiosis, diploid to haploid. To get from haploid to diploid, we're going to need 2 haploid cells, right? N plus N makes 2N. Okay, we're just joining 2 sets of chromosomes to make a full diploid set. Okay, I know this is all weird and alien. You just kind of got to remember back to mitosis and meiosis. Don't think about that too much. We're not worried about the process of mitosis or meiosis. You just have to remember the big picture. Meiosis halves, cuts the chromosomes in half, and then fertilization fuses 2 halves into a whole individual. Okay, so if you didn't get 10 out of 10 on the pre-lab, just stare at this longer and relate these words to those questions, and then that'll help you in the long run. This is your reference life cycle for liverworts. So I know we kind of think of mosses as the main seedless avascular plant, but liverworts are like our model organism. Okay, we know a lot about the liverwort life cycle, so that's what we're going to focus on, and you're going to have to identify all these different structures. So these are pictures that are labeled, and you want to relate these labeled pictures to the models. We have 3D plastic models and then images. Okay, so you can see this model is a model of this Marcantia thallus. So Marcantia is the genus of this liverwort. The liverwort because these lobed leafy structures, the thallus has kind of a liver lobe-like structure. Okay, that's what people thought it looked like, so they called it a liverwort. Okay, so we look at that life cycle. There's an asexual life cycle and there's a sexual life cycle. So if they want to just make more thallus without doing any sexual reproduction, these little Gemme cups do that. Okay, here's a Gemme cup on this. This is involved in asexual reproduction. And we can look at it under the microscope and you can see these little gymae that are going to go and become another thallus, which is like a copy. Okay, similar to how plants can grow from cuttings. Okay, you don't have to grow all seed plants from seed. You can grow from like just part of a stem can grow into a new plant. Plants are weird like that. Okay, so asexual reproduction, yeah, that's fine. They can do that, and this is the structure that helps them do that. So just relate the pictures together. The gymae cup is, this is from the side. This is a slice under the microscope. This is a whole 3D one from above. Okay. So like we said, same as mosses, the main part of the plant, the body of the plant, this is the gametophyte generation. Okay. See here, this leafy looking stuff. It's not a true leaf, but it is the photosynthetic little solar panel. This is the gametophyte. Okay, gametophyte. This is the haploid stage. Okay, the only diploid phase is the sporophyte itself. Okay, it's this little microscopic structure that lives inside of these little structures, the archegonia. Okay, so let's go here. This is asexual reproduction. And now let's look at those sexual structures. So these weird little umbrella and weird palm tree things, these are the structures that grow on the gametophyte and that produce the gametes. Okay, so we have sperm and eggs. So we're going to have separate structures to produce the sperm and eggs. Okay, so let's look here. We have the archegonia. That's this weird kind of wacky palm tree situation. That's going to be where the eggs are formed. And then we have this sort of umbrella mushroom looking thing. And that's where the sperm are going to be produced. Okay, so this is the labeled image. You need to take your time labeling these things. Okay, so these are all related. Okay, this is a microscope picture of a little antheridophore that's growing from the gametophyte. This is where the sperm are produced. The sperm are produced in these little tiny structures called the antheridia. So these are inside of this whole mushroom shape. Okay, so the 3D model, the whole model is the antheridophore, long word. The antheridia, each one of these little pockets is an antheridium. Okay, so the antheridium, here's this one, is one of them by itself. Okay, all these cells in here produce sperm. Okay, they're just doing mitosis. So sperm are going to be produced here. And you can see there's a little exit out the top. The sperm can be released through here. They fall down and then they swim over and travel to the archegoniophore. Okay, sperm are motile. Okay, so the next thing are the archegoniophores, and they have this like wacky palm tree. So they're really, really tiny. You have to look really hard for these. You can see some of them are droopy and some of them are kind of more lifted up. Okay, here's one that's droopy. These are droopy. This one's sort of more spread out. Okay, they look like little alien tree things. Now we're gonna look at them close up. Okay, so here's one of those wacky palm tree things under the microscope. When it's droopy like that, it's unfertilized. Okay, it's hanging down. That's regular archegoniophore making an egg cell. Okay, so the ovule develops inside these little archegonia. So the whole thing is an archegoniophore, and it contains archegonia. And inside each archegonium, we produce an egg cell or an ovule. Okay, so the ovule just stays there and the sperm have to come to it. Okay, so the sperm are going to kind of like swim through moisture and fertilize the egg. Once the egg or the ovule gets fertilized, then it becomes the sporophyte. Okay, so the sporophyte doesn't go anywhere else. The egg and sperm don't leave. Its fertilization happens and it grows into this little light bulb structure here. And that's our sporophyte. So underneath here are all these little sporophytes. Okay, the whole thing is still an archegoniophore. The whole wacky palm tree thing is the archegoniophore, but instead of archegonium, now we have mature sporophytes. It's weird to call it a mature sporophyte because it's just this microscopic, tiny, tiny structure. But that's it. This is the whole liverwort sporophyte. It's not big, not really visible to the naked eye. It's tucked underneath this archegoniophore. And what does a sporophyte do? Produces spores. Very good. Sporophyte produces spores. Okay, and the part of the sporophyte that produces spores is called the sporangia, which is all up in here. Okay, so you're relating this to our little life cycle diagram. We have the sporophyte generation. All it is this little light bulb shaped microscopic thing. It's not around for very long. All it does is start doing meiosis inside here. These sporangia do meiosis and they produce spores, and those spores get dumped and they go off to grow up into new gametophytes. Okay, so that's kind of putting it all together. That's the life cycle. The sporophyte generation is really brief and tiny and small and not that important, not that big of a plant. The gametophyte is kind of the main plant, and that's going to change as we move into ferns. This is confusing, okay? This just says moss gametophyte, okay? But is it this picture? Is it this picture? This is the gametophyte, okay? The gametophyte is in a moss, similar to a liverwort, this low to the ground, mossy looking thing. That's the moss gametophyte, okay? Down here, this is the moss gametophyte. And these tall little things that have like kind of a little head, those are the sporophytes. Okay, so it's a little bit more interesting than the Marcanthia sporophytes. These are actually like, they grow on a little stalk. They're kind of cute, okay? These are also sporophytes. So it's a little stalk, and then the Antheridia and the Archegonia are gonna grow inside these structures, okay? So it's a little different than the liverworts, but kind of same general idea. You don't have those little palm tree archegoniophores or antheridiophores. We just have these little fun sporophytes. So if you have moss around, you know, it's really easy to miss the sporophytes. They might just grow and go away in one day. Okay, they grow up, produce spores, meiosis produces the spores, and the spores are released, and then the sporophytes kind of die or wither away. Okay. So just be sure that you know that the stalk things, those are the sporophytes. Okay, moving into ferns, we have a more dominant sporophyte phase. So these are your ferns. This is a regular mature fern that you're used to seeing. These are what you see around. The gametophyte is a tiny, tiny little heart-shaped, leafy-like structure that's low to the ground. It's basically like a little flat leaf. You'd have to really look hard to find one. And that's the only haploid structure, okay? The ferns that we're used to seeing produce spores, and you've probably seen this. If you go look at a fern outside, you can probably find some that if you flip them over, you'll see these little dots, okay? And each little dot here on the back of the leaf is called a sorus, and that's like a little case that contains the spore producing structures, the sporangia. Okay, so the whole structure is called a sorus. That is a fern specific term. We don't have sori or sorus in mosses. This is a fern thing. And we have this little case. If we look at it under the microscope, so this is a cross section of the leaf, we can see the sporangia in here. That's where meiosis happens, and that produces the spores. Okay, so sporophyte is diploid. All this tissue up here of the leaf, these are diploid cells, and the sporangia do meiosis to produce haploid spores. Okay, so here's a little tiny gametophyte. Okay, each little spore will land in some dirt and grow into this little heart-shaped thing, and that's going to produce gametes. Okay, if we zoom in on the little heart-shaped leaf, it's got two separate areas where it produces eggs and sperm. Okay, so we've got our archegonia more centrally located and they're bigger. Okay, that's pretty typical. Things that produce eggs are usually bigger. Things that produce sperm are usually smaller. Okay, sperm are cheap. It's easy to make a ton of them because they have to scatter and go try to find the eggs. Eggs have more investment. They just hang around waiting for the sperm. So you've got your eggs here made in archegonia, same words as before. And then the antheridia are kind of off to the side, these smaller dots on the bottom sides of the heart. Okay, so similar to mosses, the sperm that are produced in the antheridia do need to swim over there. Okay, they need to swim to the archegonia to fertilize those eggs. They need a little bit of moisture. Okay, morning dew can be enough. Okay, they don't need to be in a puddle, but they do need some moisture in order to reproduce. So ferns are in many ways more adapted to land, but in terms of reproduction, they're still requiring some moisture to get the sperm to the eggs. Next week, when we meet the seed plants, we'll talk about adaptations that they can completely travel through the air to fertilize eggs. Okay, so this gametophyte is called a prothallium. So thallus, it's sort of like a phallus in a moss, but it's a prothallium in ferns. Okay, so just be able to identify these things. And these are some things that you have to label and arrange on the life cycle diagram for the exercise. Okay, here's the cool adult sporophyte. Okay, we can see a little baby sporophyte growing here. That's called a fiddlehead. They really do just unroll. Some ferns are more complex and they can roll out to the sides as well as up and down. So yeah, next time you walk past a fern, try and look for these little fiddleheads. If you've ever grown ferns, you know that they kind of just grow in a mass. Okay, and it's not necessarily one individual fern separate from the other. They're kind of all connected by this root system called a rhizome. Okay, so rhizome connects a bunch of above ground sporophytes. And then down below, we have true roots. Okay, that's a new adaptation. Also, these stems and these leaves are all new adaptations that ferns have that mosses and liverworts don't have. Okay, so true leaves have vascular tissue that collect the sugars that are being made by photosynthesis, and they transport it to the rest of the plant. Okay, true roots have vascular tissue to suck water and minerals up from the soil and send it up through the stems. So without vascular tissue, plants wouldn't be able to stand up and become this tall. Okay, so be able to label these kinds of things, keep it in order, seedless avascular versus seedless vascular. And then there are some fern friends. Okay, we've got horsetail ferns. They're very weird. We're not worried about their life cycle as much, but they are related to ferns. Instead of having photosynthetic leaves, they have photosynthetic stems. So the stems are green because that's where the chloroplasts, the cells that have chloroplasts are. And the leaves are instead reduced to this little sheath thing around each node. Okay, club mosses, they're called moss, but they're not. Okay, they're actually more related to ferns, and they do have true vascular tissue, and they have a different kind of leaf. Okay, so a true leaf, like the kind ferns have, we call them euphylls. So ferns, seed plants going forward, they all have true leaves where they have a central vein and then little branches off of that. Okay, and then okay, that's a true leaf. Okay, these club mosses or lycophytes, they just have a lycophyll that's just got one vein per leaf. Okay, no like network. Okay, these are both leaves. They work the same. They're like solar panels to collect light, but they don't, they are evolved separately. They're more analogous than they are homologous, okay, because they arise independently in different lineages, which is kind of interesting. So leaves happened more than once, but these true leaves are what ferns have and what all the plants we're going to go meet next week will have, okay? So this is a sporophyte. It's doing photosynthesis. The gametophyte's tiny and small and not doing photosynthesis. Okay, so that's a trend we're going to see keep going when we get to seed plants. Okay, so that should help you get through that life cycle activity, just going through these pictures one by one and making sure you understand where they fit in on the whole plant life cycle. This diagram, you know, like what pictures are archegonia, what pictures are antheridium, you know, zygote, sporophyte, sporangium, what produces spores. Okay, so for ferns or liverworts and ferns, you're going to put all the pictures on this life cycle and kind of like label them. Okay, so try and do that as clearly as possible, because understanding that is going to help you on the test. All right, let's take another break and keep working. If you still need to do the pre-lab, try and get that done and start working on this life cycle assignment, so that if you have problems, we can talk about it again at 9. And at 9, we'll talk about any questions you have from the seedless plants, and then we'll also kind of introduce the population genetics topic. And I'm going to have a little survey that you all need to complete by Friday so that we can do a little class analysis of population genetics, as our class is the population. Okay, so I'm gonna let y'all keep working. If you have any questions, now's the time to ask. Otherwise, I'll open the breakout rooms again and we'll check in again at 9. Does anyone have any questions to start off with? We have 25 seconds before everyone gets back from the breakout room. So the floor is open. Anything that's giving you trouble or is confusing, or you want me to talk about it again, let me know. Otherwise, I'm gonna take a look at this. 3, 2, 1. Okay, now everyone's back in here and connected. Hello, welcome back. So I'm going to switch gears. If you do have plant questions, we can talk about that more, but let me switch gears real quick and talk about population genetics. So in the announcement that I posted yesterday or the day before, it talked a little bit about this. This is a standalone module. Okay, we're not going to be tested on this. This is something that you either will or already have covered in lecture. So Hardy-Weinberg, population genetics, that's something you'll be tested on in lecture. And we have this little lab activity to go along with that, but it's not going to be on test 2. Test 2 is just plants and fungi. Okay, but you do have to get through this module, but it's kind of fun. It has these activities that sort of demonstrate what's going on with population genetics. It's like a little population genetics analysis that we're going to use our phenotype data to get through. Okay, so there's some vocabulary words you need to remember from Bio 1, and then some new words that we're kind of adding in. But all of that's kind of in this pre-lab handout, and there are videos to help you. So I have these videos here. They go in order, so you don't have to watch all of the videos first. But if you look at this part in the module, these are all my lecture videos. So if you just need more explanation, like all the nitty-gritty details, you can start there. But this first video helps with the pre-lab quiz math. Okay, so it gives you the same kind of information that you'll find in the pre-lab, and then use the Hardy-Weinberg equation to get the different allele frequencies. It seems complicated, and if you don't like math, you just have to kind of suffer through it. But once you get used to like relating what the symbols are in words, like allele frequency, recessive phenotype frequency, recessive homozygous recessive genotype frequency, these all have different symbols. And once you know which symbols go with which words, it's easy to plug in the numbers and just solve the equation. Okay. So there's a video that goes slowly through that, but I'm happy to do these problems with you. And that's, yeah, happy to do that. Okay, but these videos are there for you to like go at your own pace. When you're ready to do some math, do that. So the pre-lab quiz, you have until tomorrow to try it once. If you have a lot of trouble with it, I do have office hours tomorrow. But you know, submit it, and then if there's errors, I'll give you some feedback and maybe some pointers, and then you'll be able to try again. Okay, so it's a regular pre-lab assignment that you just should be able to get a 10 out of 10 eventually. Okay, so just do at least one attempt, try and get through this by tomorrow. The next thing that's already available to you, see it's green, it's already published. You can't see these other things because they're not available yet, because I need to collect your phenotype data. Okay, so phenotype, that's what your traits, that's all the traits you have. So like hair color, that's a character, and my phenotype for hair color is brown. Okay, I have brown eyes, my phenotype is brown eyes. So phenotype is just what you see, and then genotype is what's inside, what alleles, what genes you have. That's your instructions, and then phenotype is what's expressed. So we're going to collect for a couple of different traits. We're going to get class data, and it's just a little survey. If you do it, you'll get all the points. And it just asks, it's like 6 or 7 different traits. Okay, mid-digit hair, can you roll your tongue? Do you have a widow's peak? I always have to check, I don't really know. Earlobes, are they free or attached? And this is all explained in your pre-lab handout and the exercise handout. But just answer these questions. If you need to Google it for a comparison, you can. Once I have everyone's data, we'll analyze that data, and we'll take a look at our phenotype data, and we're going to plug it into this worksheet. So this worksheet's not available yet, but it goes through these different traits. And then we're going to say that the allele frequencies are equal. We're going to say the dominant allele for each trait. So each trait has a dominant and a recessive version. Okay, like for example, hitchhiker's thumb. You zoom in so you can read. Okay, hitchhiker's thumb, the ability to bend the thumb backwards more than 45 degrees. Okay, so I can do that. So I have a hitchhiker's thumb. That is the dominant allele. Okay, if you can't do that bending backwards thing, that's recessive. You have the recessive allele. Okay. That's the kind of data we're collecting. Then we're going to analyze our class as a population, and we're going to see if our class allele frequencies match up with our Hardy-Weinberg assumption. Okay, so it's just like a little example of what you could do with a real population, or what you could, like actual data you could collect for a real population, like a whole population. You would sample the population and then decide based on those numbers that you get if the population is in Hardy-Weinberg equilibrium. Okay, most populations are not necessarily going to be in Hardy-Weinberg equilibrium. If they're not, that usually means that that trait could be evolving. There could be some selection at play, or there could be some kind of non-random mating. Maybe certain traits are more adaptive or sexier, so they would be present in higher proportions than expected. Okay, So we have these expected frequencies for this activity. We're just saying we're expecting 50-50. 50% of the alleles in the population are dominant, 50% are recessive. Okay, so the first thing you have to do is this little chart. Okay, so we've already told you, if you read the instructions, it says you're going to start with those frequencies. Okay, so in the Hardy-Weinberg equation, we have p squared plus 2pq plus q squared, and we also have the simpler p plus q equals 1. Okay, this has to do with the recessive alleles. The alleles themselves. Allele frequencies p and q. Okay, so p is the dominant allele, q is the recessive allele frequency. That means the proportion. Okay, so we've been told that the expected frequencies are 50-50. 50% of the alleles in the population are dominant, 50% are recessive. So you could write that as 0.5. Okay, and it's the same for both, right? 0.5 plus 0.5 is what? That's 1. Okay, 50%, 50% makes 100%. That makes our whole population. That's where the 1 comes from. That means 100. Okay. When we get into genotype, okay, that's when you actually put the alleles into an individual, right? And you have to remember that dominant and recessive stuff. So let's say we have dominant allele R. I'm just going to use different letters to talk about the alleles themselves. And then little r will be the recessive allele. Now, those are the alleles on their own. What are the genotypes that you can get? There's different combinations, right? You can be big R, big R. Okay, if this is all crazy new to you, or you don't remember this from Biology 1, then go through those videos that have the background information. It's okay if you haven't covered this part in lecture yet, or if you did it for the first test, it doesn't matter. This doesn't have to go hand in hand, okay? But everyone might have different, confidence in this or different feelings about this. So this homozygous dominant, that's big R, big R. You have 2 copies of the dominant allele. Okay, so that's the genotype itself. The frequency is what this equation is about. Okay, so we have heterozygote, 1 big and 1 dominant and 1 recessive, and then we have our homozygous recessive genotype. So you just have to relate the terms to these different things and then calculate it, plug in these numbers for P and Q, right? So homozygous dominant genotype frequency, that's P squared. So what is P squared? That's 0.5 times 0.5. Okay, and then you would put that answer here. Okay, so you need to go through the pre-lab handout. It explains all these terms, and you're just going to fill in this chart. So that's like something to start with. That gives you your expected frequencies. If everything is in Hardy-Weinberg equilibrium, these are the frequencies you're going to get. Okay, so that's the little first chart. That's pretty straightforward once you just settle in. The fun thing is this chart here. Okay, so this has some old class data, but what you get here, what you're going to start with is just a blank sheet. It has this example already done for you. Oh, hey. What? When you click on this, oh geez, the computer is not liking this. I think, okay, there we go. So let me just go up to that example one. So when I collect all the class data, I'm going to give you all of the data. So for example, if I had, this is what it looks like blank, okay? You are going to fill in this column, the number of students that had a tail, the number of students that did not have a tail. Okay, this is not a trait I'm going to ask you because probably no one has a tail. Hey, come back. Okay. Stay. Okay, so you would, let's just say we have a 50-person class, okay? And let's say 32 of those students had a tail. What did I say, 50? 18? Yeah, is that, yeah, okay, good. So that calculates everything for you. All those frequencies based on this class number. So these are the actual counts of students with tails, students without tails, and the total. And based on that, it calculates the frequencies for you. Okay, so this is the class data frequencies for the trait tails. Okay, 64% of students had a tail, 36 did not have a tail. And we're saying tail is dominant, no tail is recessive. Okay, so that's the actual data for our class in this example. And then this is the expected, okay? And you will see that these numbers match the expected frequencies that you calculated up above, okay? So genotype, phenotype, this is phenotype frequencies. So the dominant phenotype is having a tail, recessive phenotype is not having a tail, okay? So you would expect 75% to have a tail, 25% to not have a tail based on a 50-50 allele spread. Okay, so this is our assumption of what should happen if everything is in equilibrium. And then we compare that to our real numbers, and this spreadsheet calculates a statistic. Okay, this statistic is how we determine whether or not our numbers are in alignment with the Hardy-Weinberg equilibrium. Okay, instead of just eyeballing it and saying, well, you know, 64% is pretty close to 75%. So yeah, we can use the statistic to determine. And the way you evaluate the statistic, we just set this arbitrary number that the p-value has to be less than 0.05 to be considered significant. Okay, and in this case, we're saying significantly different than expected. Okay, so if it's smaller than 0.05, you have a trait that's not in Hardy-Weinberg equilibrium. So this example, the p-value is 0.07. Is that less than 0.05 or greater than 0.05?
.07 is higher than 0.05.
Exactly, it's greater than. So that means it's not significantly different than expected. This is good enough. Based on our numbers, we can say we can't reject this null hypothesis that this trait is in Hardy-Weinberg equilibrium. Okay, so that's what you're going to do. So you're going to do this for all the traits. Once you all fill in your surveys, I'll give you your class data for everyone. It'll be anonymous. You won't know who has tongue rolling abilities, but we'll just say, you know, however many students can, you know, have this and don't, and you plug in the numbers, it calculates everything for you. And then you go through and find all of the traits that are significantly different, and then you're going to answer the questions below. So if it's less than 0.05, that trait is not in Hardy-Weinberg equilibrium. And that's how you're going to evaluate these things. So there's a little bit more to it, but that's just, I just want to show you how to like use this chart. If doing this in Word gives you trouble, like it was giving me, there's going to be a separate Excel sheet that you can open and do it and like use that to calculate it. That might be a little bit easier. Sometimes Excel charts in Word don't work as well. Okay, so there's going to be like 2 ways to do it. But I'm not going to give you this exercise sheet until I have all the class data. So everyone needs to get that done by Friday. Okay, so I'm going to stop there. I'm going to let y'all kind of work on that module and the seedless plants module kind of on your own. You've got time to like work your way through it. It takes some time. But if you have any questions, we have time left in the class to answer them, to go over anything you want to talk about. Next week we'll do seed plants, and then that will be a little bit more exciting because that's when we get into things like pine trees and cycads and flowering plants and all the things we eat. Yeah, really exciting plants. And then we'll get into fungi, which are really weird, and then it'll be test 2. Okay, so any questions? All right, I don't hear any questions, so I will let y'all continue working. If you are ready to work on your own, we don't have another check-in. I'll be here until 10. I'll leave the breakout rooms open. I saw that almost everyone turned in the pre-lab. That's awesome. I'll give you feedback on that. I've seen some people already retake it and get 10 out of 10, so that's really awesome. And I saw that some of the life cycle activities are already turned in, so I'll get those graded to you before next class. So if there's questions you have, like burning questions about your life cycle assignment, maybe ask about that now and we can go over it, or come to office hours. But you'll get like official feedback by next week because I want everyone to get a chance to turn it in before I give answers back. But if there's something that needs to be changed, I'll give you a chance to resubmit and get all the points if you missed any points. But that's it. Looks good so far. I'm very proud of y'all. Y'all are doing a really good job. So I'll just say good night now, but if you want to hang out until 10, you know, we've got 40 more minutes. We can keep working together. I'll open breakout rooms if you want to work together. And that is it for tonight. I'll have those test 1s graded for you ASAP. Okay, I'm going to stop recording.